Prophase 1 is the longest and most significant phase of meiosis, consuming 90% of the time for the two divisions. In prophase 1, the chromatin condenses and condenses more and more. The coils get denser and denser, tighter and tighter, and shorter, so the chromosomes become visible under a microscope. The two pairs of centrioles form and move toward the poles, forming meiotic spindle. The different chromosomes can pair up laterally with each other and are in synapses to form tetrads. Tetrads are made up of 4 chromatids, the 2 homologues, and each paired with their sister chromatid. Crossing over occurs. These are basically genetic material is swapped. This is the process responsible for creating genetic diversity by creating 4 unique chromatids. Occasionally, the gene on the chromosome that is predominantly from the mother will have some father parts in it too. During prophase 1, the nucleolus disappears. At the end of prophase 1, the nuclear envelope dissolves, allowing the spindle to enter the nucleus. Before the start of metaphase 1, crossing over is complete. In metaphase 1, microtubules grow from the centrioles and attach to the centromeres. In chromosomes, tetrads line up with each other in the middle of the cell. The metaphase plate, or the cell equator, in homologous pairs have on one pole and half on the other. In preparation to split, bivalents from either parent can align on either side of the cell. The spindle fibers attach onto the centromeres of each chromosome. Kinetochores, the proteins on the chromatids where the spindle fibers attach of each sister chromatid pair, are turned toward the same pole, causing them to attach to spindle fibers from the same pole. The chromosomes are preparing to split, which will happen in the next phase. During anaphase 1, the spindle fibers begin to pull the homologous chromosomes, or tetrads, toward opposite poles of the cell. In this phase, unless crossing over occurs, each homologous chromosome consists of 2 copies of the same set of genetic information, so they are haploid. Once this stage is complete, each side of the cell will have 23 pairs of chromosomes, and the cell will get ready to divide. Telophase 1 of meiosis is similar to telophase in mitosis. The chromosomes reach the poles of the cell. The spindle fibers and the centrioles begin to disassemble. Nuclear membranes reform around the 2 new cells, and the chromosomes fray back into chromatin. A crease forms between the 2 cells called the cleavage furrow, and the 2 new cells move in opposite directions. And as the cell membrane pinches off, 2 new daughter cells are formed that are ready to go under meiosis 2.
Prophase 2. After telophase 1, there is an intermediary stage called interkinesis. During interkinesis, the 2 cells formed during telophase 1 split, and only after the split occurs, prophase 2 can start. There are 2 stages of meiosis, with prophase 2 beginning the second part of meiosis. But there are a few differences between prophase 1 and 2. In prophase 2, the chromosomes are attached to the centrioles by spindles, while in the first stage, there are no centrioles or spindles. Also, there is no crossing over between the chromosomes, unlike in the first prophase stage. In addition, prophase 2 has a total of 2 cells originating from the cell at the beginning of prophase 1. In prophase 2, centrioles form and move to the poles of the cell. Also, the spindle fiber apparatus forms and the nuclear envelope dissolves. Only after this, metaphase 2 can start. Metaphase 2. In metaphase 2, each of the daughter cells complete the formation of the spindle apparatus. Single chromosomes align on the equator of the cell, much as chromosomes do in mitosis. This is in contrast to metaphase 1, in which homologous pairs of chromosomes align on the equator of the cell. For each chromosome, the centromere of the sister chromatids face the opposite poles, and each is attached to a microtubule coming from that pole. After this, anaphase stage 2 starts.
In anaphase 2, sister chromatids separate to become daughter chromosomes. Daughter chromosomes are moved toward opposite poles by the spindle fibers. Anaphase 2 differs from anaphase 1 due to the fact that this time, once the cells are split, each new cell contains only one set of DNA. During telophase 2, two new nuclear membranes form, one around each set of daughter chromosomes. Cytokinesis separates the two membranes. Now there are 3 chromosomes found in each cell. By the end of this, there should be 4 cells, and each cell is now haploid. This differs from telophase 1 because by the end, we end up with 4 cells instead of 2. And the final product is ERM.